One by one now we are going to consider the steps in biosynthetic phase. As you know by now that the first step was the carboxylation. Because we are dealing with that stage of photosynthesis where there would be reduction of carbon dioxide basically, that is the Calvin cycle that we were talking about. In the Calvin cycle we had three phases, not in just Kelvin cycle, there all the cycles we are having three phases, and as we talked about, whether it is C3, whether it is C4 or whether it is CAM, the main cycle that is responsible for photosynthetic fixation of carbon dioxide, that is reduction of carbon dioxide, that cycle happens to be Calvin cycle. That is studied under three stages, and we are going to take the first stage that involves the carboxylation. Okay, now what you see over here is that there is a simple equation represented where we have a compound that gets carbon dioxide fixed to itself and forms another compound. And as you can see, this number 2 numerical representation shows that two molecules are formed as a result of this reaction. Now, as you know that the carboxylation reaction is to take place, and what we have is we have a 3 carbon compound that is present over here, a 3 carbon 3 phosphoglyceric acid. That this phosphoglyceric acid has 3 carbons present inside it, and there are 2 molecules. So it is quite certain that one carbon dioxide would have combined with a molecule of RuBp which has 5 carbons. Then only we are having 6 carbons in total, and these two molecules are having 3 carbons each. That is how we equate the carbons. This is 5 carbon compound, this is single carbon compound as you can see, and we have a 6 carbon 3 carbon compounds that we have over here. So in a way we are equating the number of carbons in this equation. Now earlier studies, somehow it was studied in the beginning that the first stable outcome of the photosynthesis in the biosynthetic phase happens to be a 3 carbon compound, that is 3 phosphoglyceric acid. You have to remember this term. Why I am not starting with the cycle in the first, so that you understand what actually is happening, and when we do the graphical representation it is easy for you to understand. 3 phosphoglyceric acid, I take P, I take G from here and I take A and make it 3PGA. This is a 3 carbon compound that is being formed once the carbon dioxide gets fixed to the carbon compound, whichever it was there. In the beginning, you see what techniques were being used. They were using radioactive carbon, and the first outcome that was radioactive in the photosynthetic phase happened to be a 3 carbon compound, that is 3PGA. So for quite a long time it was assumed that the carbon compound with which it must have combined, the carbon dioxide with which it must have been combined, that must be a 2 carbon compound. You can relate with it with an idea that supposedly you have radio labeled a carbon dioxide compound, it forms a 3 carbon compound as the first outcome. It is bound to be understood that it must have combined with a 2 carbon compound, then only we are having a 3 carbon compound outcome. As you can see over here, supposedly I am getting a 3 carbon with 1 carbon CO2. It is quite certain from your knowledge of equations in mathematics, it must be 2 carbon compound. Alright, then only it would equate to 3 carbon compounds. But later on with studies, it was established that this is not a 2 carbon compound that combines with 1 carbon dioxide. Rather, it is 5 carbon compound that combines with 1 carbon dioxide. It forms an intermediate, a reactive intermediate in the beginning, and it leads to formation of 2 compounds which are having 3 carbons. This was established later on. Why it was established, how it was established, not why it was established, how it was established. This reaction is quite simple. Over here we have an enzyme that is known as Rubisco. You have to remember this term. Now let's talk a little bit about Rubisco, what it is all about. Rubisco is RuBp carboxylase. You have to remember this. This enzyme is responsible for carboxylation of RuBp. Now what is RuBp? RuBp is a sugar that is 5 carbon ribulose 1,5 Bisphosphate is the name of this sugar. Please remember. The first thing that is present inside the stroma that comes to take up the carbon dioxide is a 5-carbon compound known as ribulose 1,5-bisphosphate. In the presence of Rubisco enzyme, it combines with single molecule of carbon dioxide and forms 2 molecules of 3-phosphoglyceric acid, which is the first stable outcome of the photosynthesis, first stable compound. Though there is an intermediate formation in the beginning, that is a carboxy keto compound, but later on in the presence of water, remember this as well, in the presence of water, the intermediate compound, which is a carboxy keto compound, it breaks down to form 3-carbon compound, that is 3-phosphoglyceric acid, and 2 molecules are formed because the water, when water comes into the scenario, it breaks down that carboxy keto compound, which is a 6-carbon compound, and we have 2 molecules of 3-PGA that are formed. Now, to end up the topic, I would bring to your notice a few characteristics of Rubisco. Rubisco is the enzyme which is responsible for the carboxylation process. It is Rubp carboxylase. Remember it. Rubp, that was the compound, which is a 5-carbon compound, and the name says carboxylase. Carboxylase, ase means it must be an enzyme. Carboxylase means that enzyme which would be responsible for the carboxylation of Rubp. Otherwise, there is another component of Rubisco that is showing oxygenase. That is why we have CO in the end of this name, Rubisco. This C denotes carboxylase, this O denotes oxygenase. It is responsible for both oxygenation and carboxylation, but over here we are concerned with carboxylase activity of Rubp, and it is Rubisco, called as Rubisco. It is the most abundant enzyme in the entire world. You have to remember this thing, that if ever you find an enzyme which is most abundant in the entire world on earth, if we are talking about, it would be Rubisco. 16%, as much as 16% of enzyme of chloroplast is Rubisco, not Rubisco. 16% of the enzymes of chloroplast is Rubisco. It is present on outer surface of thylakoids. Not surface, I would say outer membrane. Present near the outer membrane of thylakoids. You can use the term surface as well, because all the reactions have to take place inside the stroma, not in the thylakoid. So, outer membrane of thylakoids, the surface of that has abundant Rubisco. As much as 16% of chloroplast enzymes are Rubisco. This Rubisco has the carboxylase activity as well as oxygenase activity. Because we are dealing with the biosynthetic phase, so we talk about its carboxylase activity, where it is responsible for fixing carbon dioxide into Rubp, and it leads to formation of 2 compounds of 3-PGA, which in the beginning, intermediate that was formed was a 6-carbon, but because of the presence of water, it got broken down into 2. So this is what happens when the carbon dioxide molecule, it gets fixed in the photosynthesis. Do remember, as expected, it should have been a 2-carbon compound with which carbon dioxide combines, but instead it is a 5-carbon compound. What is the name? Name is Rubp. What is the outcome? 3-phosphoglyceric acid, and not single molecule, it is 2 molecules of 3-phosphoglyceric acid. Do keep in mind that you have to balance the number of carbons on both sides of equations, and this is all about carboxylation.